Welcome to BioBits and Bytes. Today, we're really going to dive deep into something critical happening, well, everywhere, really. In labs and hospitals, it's the fight against viral infections.
It's a constant battle.
It really is. And, treating viruses is tough anyway, but maybe the biggest hurdle, the thing we keep running up against, is how incredibly good viruses are at developing resistance. They learn to fight back against the drugs we use.
That's absolutely spot on. And this isn't just some theoretical problem in a textbook. Antiviral drug resistance, it has really severe real-world impacts, especially, for patients who are already quite vulnerable.
Right. But when a virus gets resistant, the treatment that was working suddenly just doesn't. Or maybe not as well.
Exactly. It can become less effective, sometimes completely ineffective. And when we talk about vulnerable patients, who are we really focusing on here?
Yeah, who are the folks most at risk?
We see this pop up most often in patients whose immune systems are, weakened. Think about people who've had, say, a solid organ transplant. They receive intensive immunosuppression, right? Their immune system is deliberately dialed back to stop organ rejection. Or patients getting hematopoietic stem cell transplants, or cancer patients undergoing certain therapies that also suppress immunity.
Okay, so their body's natural defenses are sort of on the sideline.
Precisely. And that situation, it creates almost a perfect storm for resistance to develop. There are usually two key factors. First, these patients often need antiviral drugs for a long time, either to prevent an infection or manage one long-term, simply because their own immune system can't control the virus.
Right. They need that constant drug pressure.
Yes, that prolonged exposure. And second, because their immune response is weak, the virus can just keep replicating, keep making copies of itself.
And more copying means more chances for errors, right? For mutations.
Exactly right. Every time the virus copies its genetic material, there's a tiny chance of a mistake, a mutation. And if one of those random mutations just happens to make the virus less susceptible to the drug that's present, then that resistant version has an advantage. It starts replicating maybe faster than the non-resistant ones under drug pressure, and pretty soon it can become the dominant strain in that patient.
Wow. Okay. And the consequences you mentioned, they sound pretty serious.
They really are. It can range from the virus just continuing to multiply, you might see persistently high, or even increasing viral load in the blood, which we call viremia. Could you break down viremia quickly?
Yes, viremia. So that term just means, quite simply, the presence of virus particles circulating in the bloodstream. You can measure it with lab tests.
Perfect. So you get that persistent viremia, or the actual disease caused by the virus might get worse, even though the patient is on treatment. Then, if the first-line drug fails, we have to switch
to second-line options.
Right. And those are often less ideal. Maybe less effective, maybe more toxic, more side effects. And in the worst case scenarios, honestly, if the virus develops resistance to multiple drugs and we just run out of good options, well, it can lead to very severe disease or even be fatal.
That really paints a picture of how high the stakes are. Now, based on the research we're looking at for this deep dive, are there specific viruses that are, let's say, particularly notorious for doing this?
Yeah, there are definitely some usual suspects highlighted in the sources. For this discussion, we're going to focus on a few key ones. Cytomegalovirus, or CMV, that's a huge concern, especially after organ transplants.
Okay, CMV.
Then there's herpes simplex virus, HSV, which causes things like cold sores and genital herpes. Also, varicella zoster virus, VZV, that's the one behind chickenpox and shingles.
Right.
And finally, hepatitis B virus, HBV. Chronic HBV infection needs really long-term therapy, which, as we said, creates that perfect opportunity for resistance. These are all viruses where resistance is a major clinical headache, particularly in those immunocompromised patients.
Okay, so we get why it's such a big deal. Now, let's really get into the weeds. How do these tiny viruses actually manage this? What's happening down at the biological, the molecular level?
Yeah, fundamentally, it almost always comes down to changes in the virus's genetic instructions, its DNA, or RNA for some viruses. These changes are mutations.
Little typos in the genetic code.
Kind of, yeah. And these typos change the viral proteins that are built from those instructions. Usually the mutations affect enzymes that are absolutely essential for the virus, maybe for copying itself or interacting with the host cell, or crucially interacting with the antiviral drug.
Let's start with those herpes viruses you mentioned, HSV and VZV.
Yeah.
A common drug for them is acyclovir, right? How do they manage to become resistant to that?
Okay, so for HSV and VZV, resistance typically involves mutations in one of two key viral genes. By far, the most common place we see mutations is in the gene for an enzyme called viral thymidine kinase, or TK.
Viral thymidine kinase, TK. Okay, maybe you could unpack that for us. What does this TK enzyme actually do, and why is it so central to resistance?
Absolutely. So thymidine kinase, TK, it's an enzyme the herpes virus makes inside the cell it infects. Now, many of our best antiviral drugs for herpes viruses, acyclovir, like you mentioned,
Yeah.
also drugs like ganciclovir and penciclovir, they're actually what we call prodrugs.
Prodrugs, meaning they're not active right away.
Exactly. They're inactive when you take them. They need to be chemically modified or activated inside the infected cell to work. And for these particular drugs, the very first step in that activation process, the first chemical modification, is carried out by the viral TK enzyme. The virus itself has to kickstart the drug that's meant to stop it.
Wait, so the virus's own enzyme is needed to flip the switch on the drug designed to kill it?
Yeah.
That seems like a major vulnerability for the virus.
It absolutely is, and that's precisely why the TK gene is such a hotspot for resistance mutations. The vast majority, we're talking over 90% of acyclovir resistance found in clinical samples, is caused by mutations in this viral TK gene. The virus basically adapts by breaking or changing the very enzyme needed to turn the drug on.
Okay, so how does it break it? What kinds of changes happen in that TK gene?
Well, there are broadly two main ways this happens. The most frequent type, and the one that usually causes the biggest problems clinically, results in the virus essentially losing its TK activity altogether. The mutation might, say, delete a chunk of the gene, or introduce a signal that tells the cell to stop building the protein too early, or just mess up the protein structure so badly it's completely non-functional.
So the enzyme is just
Yeah.
gone or totally broken.
Pretty much. Now, these TK-deficient viruses, they can still replicate, maybe not quite as efficiently as the original virus sometimes, but they can manage because our own host cells have their own version of TK that the virus can sometimes use a little bit, although the host TK doesn't activate these drugs efficiently at all.
Okay, so broken or missing enzyme is the main way. What's the second type of TK mutation?
The second type is a bit less common. It involves what we call an altered TK substrate specificity. Here, the enzyme is still made, and it still has some of its normal TK function for the virus, but the mutations change its shape or its active site just enough
Uh-huh.
so that it doesn't recognize or grab onto the antiviral drug molecule effectively anymore. It can't perform that crucial first activation step on the drug, or does it very poorly.
Got it. So either the key enzyme is missing in action, or it's still there, but it's sort of warped, so the drug molecule doesn't fit properly. How do these mutations actually happen in the DNA sequence itself? What causes these changes?
Often they're just small mistakes that occur during the virus's DNA copying process, which happens really fast. For instance, sometimes a single nucleotide, one of the DNA building blocks, might get accidentally added or deleted, especially in areas where there are repetitive sequences like long strings of Gs or Cs. This can cause something called a frame shift mutation.
Frame shift mutation. Can you explain that? Sounds important.
Yeah, it's a really disruptive type of mutation. Think of the DNA sequence being read by the cell's machinery in groups of 3 letters called codons. Each codon specifies an amino acid, the building block of a protein.
Right, like 3-letter words making up the protein sentence.
Exactly. Now, if you insert or delete a number of letters that isn't a multiple of 3, say, just 1 or 2 letters, it shifts the whole reading frame from that point onwards. All the downstream codons are read incorrectly.
Ah, so if the sentence was the fat cat ate the rat, and you delete the F, it becomes the ATC ate a tet her at gibberish.
Perfect analogy. It scrambles the rest of the message completely. Usually this leads to a premature stop signal or just a totally non-functional protein.
Right.
And in the case of TK, this often means complete loss of enzyme activity.
Okay, that makes sense. And you mentioned these TK mutations often lead to cross-resistance. What does that mean?
Right. Cross-resistance means that a mutation conferring resistance to one drug also makes the virus resistant to other, often related, drugs. Since acyclovir, ganciclovir, and penciclovir all depend on that initial activation step by viral TK, if the virus knocks out its TK function, it generally becomes resistant to all 3 of those drugs at the same time. Now, sometimes those altered specificity TK mutants might retain a little bit of sensitivity to one drug over another, maybe penciclovir sometimes, but generally TK loss means broad resistance to that class.
That's very efficient resistance from the virus's point of view. You said that there's a second gene sometimes involved in HSV-VZV resistance.
Yes, although less frequently, mutations can also pop up in the gene for the viral DNA polymerase.
DNA polymerase. Okay, what's the job of that enzyme?
The DNA polymerase is absolutely fundamental. It's the main enzyme the virus uses to replicate its genetic material to make new copies of its DNA genome. It's the core copying machine. Without it, the virus can't reproduce.
So mutations here are probably less common because messing this up too much would just kill the virus.
That's likely the reason, yes. Significant changes could easily cripple the virus's ability to replicate at all, but mutations do occur.
And where in this polymerase enzyme do these resistance mutations tend to appear?
They don't just happen anywhere. They tend to cluster in specific functional areas or domains within the enzyme. Often these are regions involved in binding the DNA template or grabbing the nucleotide building blocks, or, importantly, the site where the drug molecule itself binds or interferes. Mutations here can directly reduce how well drugs that target the polymerase, like foscarnet or cidofovir, can inhibit the enzyme.
Okay, so TK mutations affect the drugs that need activating by TK, but DNA polymerase mutations affect the drugs that directly target the polymerase enzyme itself.
Correct. And that distinction is really critical when it comes to treatment decisions. Drugs that don't need that viral TK activation step, think foscarnet and cidofovir, can still work perfectly well against viruses that have lost their TK function. Foscarnet directly inhibits the polymerase, and cidofovir gets activated by our own host cell enzymes.
So they bypass the broken TK pathway.
Exactly. However, if the virus is unlucky enough, or perhaps lucky from its perspective, to develop a mutation in its DNA polymerase in addition to having a TK mutation,
Then you've got a problem.
then you can become resistant to those non-TK dependent drugs too, and that really starts to limit the treatment options available. It's like the virus putting up a second line of defense.
Right. Okay, let's shift gears to CMV, cytomegalovirus.
Yeah.
You said it's a big issue in transplant patients. Ganciclovir is a key drug there. How does CMV typically become resistant?
For CMV, the story is quite similar in principle, but involves different specific genes. The most common route to ganciclovir resistance involves mutations in a viral gene called UL97. This gene codes for a viral kinase.
UL97 kinase, so another kinase enzyme like TK. What does this one do for CMV?
Very similar role in this context. UL97 is a viral protein kinase. It adds phosphate groups to proteins, which is important for CMV's replication cycle. But critically for resistance, it's the main viral enzyme responsible for that crucial first phosphorylation step needed to activate the ganciclovir prodrug inside the infected cell.
Okay, so just like TK for HSV, CMV's UL97 kinase is needed to switch on ganciclovir. I'm definitely seeing a pattern here.
It's a very common strategy for these viruses, and just like with TK, mutations in UL97 are the dominant cause of ganciclovir resistance in CMV. Again, over 90% of resistant CMV found in patients has UL97 mutations.
And how do these mutations work? Do they just break the enzyme?
Not usually. What's clever from the virus's perspective is that these mutations typically decrease the enzyme's ability to phosphorylate ganciclovir specifically, but they often don't significantly mess up the enzyme's other essential functions for the virus. The virus manages to keep the vital parts of the enzyme working while changing the part that interacts with the drug.
Wow. It selectively disables the drug activation part. Do these mutations happen randomly in the UL97 gene, or are there specific locations?
They definitely cluster in specific hotspot regions. The most common mutations are found concentrated around codons 460 and 520, and then there's another key region between codons 590 and 607. You see the same specific mutations popping up again and again in resistant clinical samples, things like M460V, M460I, H520Q, C592G, A594V, L595S, C603W. These are the usual suspects.
And do all these different hotspot mutations cause the same amount of resistance?
No, there's variability. Most of those common ones, like in the 590-607 stretch or the M460VI, H520Q mutations, they generally confer what we'd call moderate resistance to ganciclovir. This is often measured using something called an IC50 ratio.
IC50 ratio. Okay, you mentioned that before. Can you remind us what that tells you about resistance?
Sure. The IC50 is the inhibitory concentration 50%. It's the concentration of a drug needed in a lab test to inhibit the virus's replication by 50%. The IC50 ratio compares the IC50 for the patient's potentially resistant virus to the IC50 for a standard, known susceptible, wild-type virus. So an IC50 ratio of, say, 10 means you need 10 times more drug concentration to inhibit the resistant virus by 50% compared to the susceptible one. A higher ratio means higher resistance.
Got it. So those common UL97 mutations give maybe a 5 to 10-fold increase in the IC50?
Typically in that range, yes, 5 to 10. But there are exceptions. For instance, the C592G mutation usually only confers low-level resistance, maybe an IC50 ratio around 2.5. So the specific mutation matters.
And you can actually test the effect of a specific mutation in the lab.
Yes, that's often done using a technique called recombinant phenotyping. Phenotyping just means measuring the observable characteristics, in this case, drug sensitivity. What is recombinant phenotyping exactly?
Ah, right. So recombinant phenotyping is basically an in vitro technique, meaning done in the lab in cell culture, not in a person. Scientists will take a standard susceptible lab strain of the virus, and using genetic engineering tools, they'll introduce the specific mutation, say A594V, that was found in a patient's resistant CMV isolate. They essentially recreate the patient's mutant virus in the lab. Then they grow this engineered recombinant virus and test its sensitivity to ganciclover and maybe other drugs, measuring its IC50 directly. This confirms whether that specific mutation actually causes the resistance seen and quantifies how much resistance it confers. It's a way to link the genetic change, genotype, to the functional outcome, phenotype.
Exactly. It helps validate the clinical significance of finding a particular mutation. So UL97 is the main story for ganciclover resistance in CMV. But is there a plan B for the virus, like the DNA polymerase mutations in HSV?
Yes, there is. Just like HSV, CMV can also develop resistance through mutations in its viral UL54 DNA polymerase gene. This is CMV's version of that essential DNA copying enzyme.
The main replication engine again. What happens when this gene gets hit in CMV?
Mutations in UL54 are particularly concerning because they have the potential to confer resistance not just to ganciclover, but potentially to any or even all of the main anti-CMV drugs currently available. So ganciclover, cidofovir, and foscarnet.
Wow, okay. That's serious. Does one mutation knock out all three?
Not always. The specific pattern of resistance or cross-resistance really depends on where the mutation occurs within the UL54 gene. For example, mutations in certain domains involved in proofreading, called exonuclease domains, often cause resistance to both ganciclovir and cidofovir. Other mutations, maybe closer to the core catalytic site where the DNA building blocks are added, might primarily confer resistance to foscarnet, sometimes with maybe a low level of cross-resistance to the others.
So the location of the mutation determines which drugs are affected.
Largely, yes. But here's the really worrying part. Although it's uncommon, there are specific single mutations within UL54 that can actually confer clinically significant resistance to ganciclover, cidofovir, and foscarnet all at once.
Triple resistance from one mutation. Ouch.
Exactly. These are rare, but they present a huge clinical challenge because you've lost basically all your standard licensed options.
Is it common for CMV to acquire mutations in both UL97 and UL54 over time?
Yes. Unfortunately, that sequential accumulation is quite common, especially in patients who require long courses of antiviral therapy. What often happens is the virus first develops a UL97 mutation, giving it that moderate ganciclover resistance.
Okay.
If the patient continues on ganciclover therapy, the virus is under constant pressure. Eventually, a variant might emerge that has also picked up one or more mutations in the UL54 polymerase gene on top of the existing UL97 mutation.
So it adds layers of resistance.
Precisely.
Yeah.
And this combination, UL97 plus UL54 mutations, typically leads to much higher levels of ganciclover resistance and often broad resistance to multiple drugs. It makes the infection incredibly difficult, sometimes, frankly, impossible, to clear using the standard antivirals.
That's a really clear picture of how this escalates under drug pressure. Okay, let's switch to our last example virus, hepatitis B, HBV. You mentioned it needs long-term treatment. How does resistance play out there?
Right, so for HBV, the main target for the drugs we use is, once again, the viral DNA polymerase. But the HBV polymerase is a bit special. Could you tell us about it?
Sure. The HBV DNA polymerase is quite a complex enzyme. It does the standard job of synthesizing DNA. But crucially, for HBV's unique replication cycle, it also has a reverse transcriptase activity. What does that mean?
Reverse transcriptase means it can actually synthesize a DNA strand using an RNA strand as a template. Most DNA polymerases, like our own, make DNA from a DNA template. This reverse transcription step is key to how HBV replicates its genome.
Okay, so it's a dual-function enzyme, and the drugs target this.
Exactly. The cornerstone drugs for treating chronic HBV are a class called nucleoside and nucleotide analogs.
Yeah,
These include drugs like lamivudine, adesivir, and entecavir, and tenofovir.
Hmm.
These drugs get taken up by the infected liver cells. Inside the cell, our own host enzymes activate them, converting them into a form that looks very similar to the natural DNA building block.
Like decoys.
Precisely. The viral DNA polymerase gets fooled. It grabs one of these fake building blocks and tries to incorporate it into the new viral DNA chain it's making. But usually, once the fake block is added, the chain can't be extended any further. It acts as a chain terminator, stopping viral DNA synthesis.
Okay, so they block the replication process. Why is resistance still such a big problem for HBV then, if these drugs work well?
The core issue lies in how HBV persists inside infected liver cells. It establishes a very stable form of its genome in the cell nucleus called covalently closed circular DNA, or cccDNA. Maybe you could describe cccDNA for us.
Yeah, cccDNA is really the heart of the persistence problem. When the HBV virus first infects a liver cell, its DNA genome gets transported to the nucleus. There, it's converted into this unique structure, a closed loop of DNA, like a tiny independent mini chromosome that's very stable and long-lasting.
And this cccDNA acts as the master template.
Exactly. It serves as the blueprint from which all the viral RNAs are made, and those RNAs are then used to make viral proteins and also serve as the template for the reverse transcriptase activity of the polymerase to make new viral DNA genomes. Now, here's the catch. Our current antiviral drugs, the polymerase inhibitors, are great at stopping that last step, the creation of new viral DNA from the RNA template.
Right.
But they have very little, if any, direct effect on the cccDNA that's already sitting there in the nucleus. They don't eliminate the blueprint.
Ah, so you're stopping the factory from making new products, but the factory's master plan is still safely filed away.
That's a perfect analogy. Because the source of new virus production isn't eliminated, treatment for chronic HBV has to be very long-term, often lifelong, just to keep viral replication suppressed continuously.
And that long-term drug exposure gives resistance mutations plenty of time to pop up and take over.
Precisely. The constant drug pressure selects for any pre-existing or newly arising mutations in the viral DNA polymerase gene that make the enzyme less sensitive to the drug.
Now, besides factors like how long someone is treated or their immune status, are some HBV drugs inherently more prone to resistance than others?
Yes, definitely. Two key drug characteristics really influence the risk of resistance emerging. One is the drug's potency, basically, how strongly it suppresses viral replication and reduces the amount of virus. The other crucial factor is the drug's genetic barrier to resistance.
Genetic barrier. That sounds important. What does that term mean?
Think of the genetic barrier as a measure of how hard the virus has to work, genetically speaking, to become resistant to a specific drug while still remaining functional. It relates to the number and type of specific mutations the virus needs to acquire in its polymerase gene to significantly reduce the drug's effectiveness without crippling its own ability to replicate.
Okay, so a high barrier means it needs lots of complicated mutations.
Exactly. A drug with a high genetic barrier might require multiple specific mutations, maybe in combination, to cause significant resistance. This makes resistance emergence less likely, statistically rarer. Conversely, a drug with a low genetic barrier might only need one or maybe two relatively simple mutations, making resistance much easier and more common to develop.
So ideally, you want a drug that's both highly potent and has a high genetic barrier.
That's the goal. And looking at the HBV drugs based on the sources, we see clear differences. The older drug, lamivudine, it has decent potency but a low genetic barrier. And that's reflected in really high resistance rates. The sources mention up to 70% of patients developing resistance after 5 years of starting lamivudine therapy if they were treatment naive.
70%, wow.
Well,
What about the others?
Adefovir has lower potency, but an intermediate genetic barrier, leading to intermediate resistance rates, may be around 29% at 5 years. Telbivudine is interesting. It has high potency, but unfortunately also a low genetic barrier, resulting in intermediate resistance rates again, something like 20% resistance emerging within just 2 years.
Okay, so low barrier seems to be the main issue there.
Mhm.
What about the newer mainstays, entecavir and tenofovir?
They really changed the game. Both entecavir and tenofovir are considered high-potency drugs, and they have a high genetic barrier to resistance. This makes a huge difference. For entecavir, the resistance rate in treatment-naive patients is incredibly low, only about 1% after 5 years. Tenofovir also has very low rates of resistance reported.
That's a massive improvement over lamivudine. Does a patient's previous treatment history affect resistance to these newer drugs?
Oh, absolutely. It's a major factor. If a patient has already been treated with, say, lamivudine and developed resistance to it, their risk of developing resistance to subsequent drugs like entecavir or adefovir is much, much higher. For example, if a patient with lamivudine resistance switches to entecavir, their risk of developing entecavir resistance over 5 years jumps from that low 1% up to over 50%.
Wow. So the prior resistance mutations make it easier for the virus to become resistant to the next drug.
Exactly. The virus might already have some mutations that give it a head start towards resisting the new drug, or it might only need one additional mutation on top of the old ones. It really underscores why choosing the right first-line therapy is so important and why monitoring for resistance is crucial.
Are there specific well-known HPV resistance mutations that doctors look out for?
Yes, several mutations in the polymerase gene are well characterized. For example, mutations like N236T or A181VT are known to be associated with resistance to adefovir. The specific mutation matters for cross-resistance. N236T might still allow treatment with lamivudine or telbuvudine, though maybe not for long if there was prior lamivudine resistance. But A181V can decrease the activity of lamivudine and telbuvudine. And importantly, the A181T mutation often confers cross-resistance to lamivudine, so that drug should probably be avoided, while entecavir and tenofovir usually remain effective options. Management really depends on the specific mutation profile.
Okay, we've really gone through the nitty-gritty of how these viruses chemically and genetically outsmart our drugs. It's fascinating, but also daunting.
Yeah.
They mutate these key enzymes, TK, UL97, DNA polymerase in very specific ways. And managing this clinically sounds like a real challenge, trying to figure out the mutations, dealing with drug side effects, running out of options.
It definitely paints a picture of the challenges clinicians face every day with these resistant infections.
This seems like exactly the kind of complex, data-heavy puzzle where modern technology, things like AI and machine learning, could potentially make a big difference.
I think you're absolutely right. This is precisely where fields like machine learning, artificial intelligence, and other advanced computational methods are starting to offer really exciting new tools in the broader fight against antimicrobial resistance, or AMR, which definitely includes antiviral resistance.
Okay, AI and machine learning. We hear these terms a lot. Could you give us a quick sort of working definition in this context? What are we talking about?
Sure. So artificial intelligence, AI, is the really broad concept of creating computer systems that can do tasks that normally require human intelligence, things like learning, reasoning, problem solving, making decisions. Machine learning, ML, is a major part, a subfield of AI. The key idea of ML is that we develop algorithms that allow computers to learn patterns and make predictions or decisions directly from looking at lots of data, rather than being explicitly programmed with detailed instructions for every single possible situation.
So instead of writing a giant rule book for the computer, you show it tons of examples, and it figures out the rules itself.
That's a great way to put it. And the interest in using ML for infectious diseases, especially for tackling resistance, has just exploded recently. There are a few reasons why now is the time. We have this just exponential growth in, biological and medical data we can generate, genomic sequences, clinical data, drug screening results.
Right, the data flood.
Exactly. Plus, we have massively more powerful computers than ever before, and there have been huge breakthroughs in the ML algorithms themselves, especially in areas like deep learning.
And ML is already being used in other areas of infectious disease, too.
Oh, yes, quite broadly. It's being explored for things like making diagnoses faster or more accurate, helping predict which patients might develop dangerous conditions like sepsis earlier, or figuring out the best strategies for using preventive antibiotics or antivirals. But yeah, today our focus is specifically on its role in understanding and combating AMR.
Now, the sources we looked at seem to categorize the ways ML is being applied to AMR into about three main areas.
That's right. The first big area is using ML to predict antimicrobial resistance directly from the pathogen's genetic data. So look at the DNA sequence and predict if it's going to be resistant or susceptible to a drug.
Okay, prediction from genomics.
The second area is using ML to help us understand the underlying biological mechanisms. How do these drugs actually work? How does resistance develop? And also using ML to accelerate the discovery and even the design of totally new antimicrobial drugs.
Understanding mechanisms and finding new drugs. Got it.
And the third area mentioned is applying ML to improve antimicrobial stewardship. That means using clinical data, often from electronic health records, to help doctors make better decisions about prescribing antibiotics or antivirals, optimizing their use to hopefully slow down the emergence of resistance in hospitals or communities.
Okay, prediction, understanding mechanisms, and drug discovery and stewardship. Given our earlier discussion on the complex biology and treatment challenges, and knowing we want to talk about that specific IIT-BHU framework later, focusing on those first two areas, prediction and understanding mechanisms drug discovery, seems most relevant for today's deep dive.
I agree. That makes perfect sense. They tie in really nicely with the viral resistance mechanisms we've already discussed.
Great. So let's start with prediction. How exactly does a computer look at a string of DNA letters from a virus or bacterium and figure out, this one's resistant to drug X? It can't just see the resistance mutation, right?
Chuckles slightly. No, not usually that directly. It's more about the ML models learning very complex patterns within that genetic code that consistently correlate with resistance. The whole process starts with data, lots of it. You need large data sets where you have, for each microbial sample or isolate, two key pieces of information paired together.
Okay, what are those two pieces?
First, you need its genetic sequence, maybe the whole genome, or at least the key genes known or suspected to be involved in resistance. Second, you need the result from a laboratory test that actually measured how susceptible that same isolate was to various drugs. That lab testing is called AST.
AST, could you define that for us?
Sure thing. AST stands for antimicrobial susceptibility testing. It's a standard procedure in clinical microbiology labs. They take the bacteria or virus isolated from a patient sample, grow it in the lab, and then expose it to different concentrations of relevant antimicrobial drugs. The test determines the lowest concentration of each drug that stops the microbe from growing, basically measuring how sensitive or resistant that specific strain is.
Okay, so you need the genetic blueprint, the sequence, and the lab result saying, yes, this drug worked, or no, it didn't work against that specific blueprint. That paired data is the fuel for the ML model.
Exactly. That's the training data. This enables what's called genotypic AMR prediction. The goal is to build a model that can look only at a new genetic sequence and accurately predict what the AST result would be. Will it be susceptible, intermediate, or resistant?
How does that compare to older ways of predicting resistance from genetics?
Well, the more traditional approach is rule-based. You essentially tell the computer, look for this specific known resistance gene, or look for this specific mutation at this position. This works okay for simple cases where we already know the mechanism well, but you have to manually discover and catalog all those rules, keep updating the catalog, and it struggles when resistance is caused by combinations of genes or subtle effects we don't know about yet.
So ML is different because you don't have to pre-specify all the rules.
Precisely. ML models, particularly supervised learning models, learn the relevant patterns directly from the data. They can identify combinations of genetic features, maybe across multiple genes, that are predictive of resistance, even if those features weren't previously known resistance markers. This makes ML potentially much more powerful for tackling complex resistance and scalable to new pathogens or drugs. The flip side, as we sort of hinted at, is that some ML models can be like a black box.
Right, they give you the answer, but not necessarily the why.
Yeah, the prediction might be accurate, but understanding which specific genetic features drove that prediction can sometimes be difficult with certain types of models.
We'll come back to that interpretability challenge. But first, how do you actually prepare that raw DNA sequence, that long string of A's, T's, C's, and G's, so that a mathematical ML model can work with it? You need numbers, right?
You do. You need to convert the sequence data into a structured format, usually numerical. This process is called feature extraction. Could you maybe elaborate on feature extraction in this context?
Sure. So in machine learning, feature extraction is all about transforming raw, often messy or complex data, like a genome sequence, into a set of meaningful characteristics or features that the learning algorithm can actually understand and process. You try to capture the essential information in a more structured, often numerical way.
That's a great explanation, and of a very common technique for doing this with genomic sequences, especially for AMR prediction, is something called the K-mer approach.
K-mers. Okay, what exactly is a K-mer?
A K-mer is simply a short consecutive subsequence of DNA or RNA letters of a specific length, K. So if you set K to be, say, 5, then ATGCG would be one 5-mer found within a longer sequence. TGCTT would be the next overlapping 5-mer, and so on.
Okay, so you're basically chopping the entire genome sequence up into lots of small overlapping pieces of fixed length K.
That's the essence of it, yes. You slide a window of size K along the whole sequence, one base at a time, and collect all the unique K-mers you find. Then, for each bacterial or viral isolate in your dataset, you essentially count how many times each possible K-mer appears in its genome. Or sometimes you just note the presence or absence of each chimer.
So you end up with like a massive checklist or frequency count for every possible short sequence of length k.
Exactly. It creates a very large vector, a long list of numbers, where each number represents the frequency or presence of a specific chimer. This chimer vector profile for each isolate becomes the input features for the ML model.
And this chimer data gets paired with the AST result.
Correct. You merge this chimer profile with the known phenotypic AS2 results, susceptible, resistant, the ground truth label, for that same isolate. The ML algorithm may be something like logistic regression or a decision tree, or even a complex neural network then analyzes this combined dataset. It learns to identify which specific chimers or combinations of chimers are statistically associated with resistance versus susceptibility for a particular drug.
What kind of K lengths are typically used?
It varies, but common lengths mentioned in the sources range from maybe K13 up to K31 nucleotides. There's a trade-off. Longer chimers are more specific, potentially pinpointing unique gene regions better, but there are vastly more possible long chimers, so you need much larger data sets to ensure the model sees enough examples of each potentially important long chimer to learn reliably.
It sounds like a clever way to capture genetic variation without needing to first map everything to specific genes. But getting accurate predictions must still have its challenges.
Oh, definitely. The sources are clear about several major hurdles. A really big one is simply getting enough high-quality data. There's a relative scarcity of large, publicly available databases that reliably link both the complete genomic sequence data and standardized, accurate phenotypic AST results for a wide variety of pathogens and drugs. Good training data is paramount.
So just finding enough well-documented examples of resistant strains, especially for less common bugs or drugs, is hard.
It is. And another significant challenge lies in the potential variability or noise in the phenotypic AST data itself. Remember, the ML model is learning to predict this lab result. So if the lab results used for training are inconsistent or have errors, that fundamentally limits how accurate the ML prediction can ever be. It's learning from an imperfect ground truth.
What makes those AST results variable? Aren't they standardized?
They are standardized to a degree, but there can still be variations between labs, changes in testing methods over time, differences in how clinical breakpoints are set and interpreted, missing data sometimes, and just inherent biological variability that leads to slightly different minimum inhibitory concentrations or MICs when the same strain is tested multiple times or in different labs.
Breakpoints and MICs, could you remind us what those are?
Absolutely. The minimum inhibitory concentration, or MIC, is the specific result from the AS2 test. It's the lowest concentration of the drug that visibly stops the microbe from growing in the lab. It's a quantitative measure, like 8 micrograms per milliliter. A breakpoint is a specific MIC value or threshold that's established by expert committees based on clinical and microbiological data. This threshold is used to categorize the microbe as susceptible, intermediate, or resistant to that drug for guiding treatment decisions. It translates the quantitative MIC into a clinical category.
Okay, MIC is the lab number, breakpoint is the clinical cutoff.
Exactly. And predicting the category is often hardest for isolates whose MICs fall very close to that breakpoint value, or those categorized as intermediate. For some drug-bug combinations, the MIC values for susceptible strains and resistant strains overlap quite a bit, making it hard for a model to draw a clean line. Prediction is generally easier when there's a clear separation, a bimodal distribution, between the MICs of the susceptible and resistant populations.
Right, trying to classify something right on the border is always tricky.
Yes, and some research suggests that prediction accuracy can actually be improved if these ambiguous near breakpoint or intermediate isolates are handled differently during model training, may be removed or grouped separately.
And of course, these prediction tools need to be expanded beyond just the most common infections and drugs.
Absolutely. For widespread clinical utility, we need reliable ML predictors that cover the full spectrum of pathogens and antimicrobials encountered in practice globally.
Okay, so ML prediction using genetics is powerful, definitely promising, but still faces these data and interpretation challenges.
Yeah.
And crucially, as you mentioned, it often gives you the what's resistant or susceptible, but not necessarily the why.
Exactly. That's the black box issue again. A model using chimers might be great at predicting, but those chimer patterns don't automatically tell a biologist, aha, resistance is caused by this specific mutation altering the drug binding site in this enzyme. This is why there's such a big push towards developing interpretable ML methods.
Interpretable ML, what's the goal there? To peek inside the black box.
Essentially, yes. Interpretable ML focuses on creating models and techniques that are designed not just for predictive accuracy, but also to provide meaningful insights into how they arrive at their predictions. In biology, the aim is often to connect the model's findings back to underlying biological processes, structures, or features. We want to understand the biological story, not just get a classification label.
So bridging the gap between the data patterns the computer finds and the actual biology happening in the cell.
That's the core idea. It might involve designing the model architecture differently or using post-hoc analysis techniques to identify which input features like specific chimeras, genes, or mutations were most influential in the model's decision. The goal is to make the model's reasoning transparent and biologically meaningful.
Can you give an example from the sources of how this interpretability is helping us understand resistance mechanisms better?
One really interesting approach mentioned uses network models. A study by Yang and colleagues used a metabolic network model of bacteria to study antibiotic action. Could you explain what a metabolic network model is?
Right. A metabolic network model is essentially a computational map of all the known biochemical reactions that happen inside a cell. It represents the enzymes, the molecules they convert, metabolites, and the pathways they form, how the cell takes in nutrients, generates energy, builds essential components, etc. It's like a detailed biochemical flowchart for the cell.
That's a perfect description. So Yang et al. Took experimental data on how bacteria responded to antibiotics and used this metabolic network model to translate that data into descriptions of the bacteria's overall metabolic state. They then used ML to analyze these network states and found that specific metabolic pathways, like the one for making purines, DNA building blocks, were unexpectedly important for determining whether the antibiotic could actually kill the bacteria.
Wow. So it wasn't just about the drug hitting its direct target, but about how the rest of the cell's metabolism reacted.
Exactly. It suggested that resistance or tolerance might sometimes involve changes in seemingly unrelated metabolic pathways that somehow help the cell survive the antibiotic's effects. It opens up new avenues for understanding resistance beyond just the immediate drug-target interaction. Another example used even more comprehensive models called genome-scale metabolic models, or GEMS, this time for M. tuberculosis, the TB bacterium.
Genome scale. So that's like the complete metabolic map for the entire organism.
Pretty much, yes.
Yeah.
As complete as current knowledge allows. Cavas and colleagues took this a step further. They incorporated genetic variation data, specifically SNPs, from clinical isolates of drug-resistant TB into these GMs. What are SNPs again?
SNPs are single nucleotide polymorphisms. They're the most common type of genetic variation, just a difference in a single DNA letter, A, T, C, or G, at a specific position in the genome when comparing individuals or strains. So they were looking at these single letter differences found in resistant TB strains.
Right. They plugged these SNPs from resistant bacteria into the big metabolic map. Then they used ML to analyze how these genetic changes altered the predicted activity levels of different metabolic reactions within the models. The goal was to find which metabolic reactions became predictive of resistance to various anti-TB drugs.
And what did they find? Did it just point to the known drug targets?
It did identify changes related to known drug targets, but interestingly, it also implicated other metabolic enzymes and pathways as being important drivers of resistance. Again, it highlighted that resistance can be a complex metabolic adaptation, not just a simple mutation in the target protein.
That's a powerful demonstration. ML isn't just confirming what we know, it's helping uncover potentially novel resistance mechanisms by looking at the system more holistically.
That's exactly the potential of interpretable ML in this field. It moves beyond prediction towards generating new biological hypotheses that can then be tested experimentally.
This ability to generate new biological insights is just fascinating. Can we pivot this technology now? Can it also help us find completely new ways to treat these infections, like discovering new drugs?
Yes, absolutely. ML is becoming a really important tool in the drug discovery pipeline as well. Now, it's fair to say the specific examples highlighted in our source materials focused more on discovering new antibacterials or designing antimicrobial peptides rather than antivirals directly. But the underlying principles and the computational techniques are very much applicable to finding novel antivirals too.
So the same AI methods could screen for antiviral compounds.
Precisely. The basic concept is that ML models can be trained on vast amounts of chemical data. They can learn the complex relationships between a molecule's structure and its properties, including its potential biological activity. Then, these trained models can be used to rapidly screen enormous digital libraries databases containing structures of millions, even billions, of chemical compounds.
To pick out the needles in the haystack.
Exactly. The model can predict which molecules in that library are most likely to, say, bind effectively to a specific viral protein target, inhibit a key viral enzyme, or generally show antiviral activity. This allows researchers to computationally sift through chemical spaces that would be completely impossible to test physically in the lab and prioritize a much smaller, manageable number of promising candidates for actual experimental testing.
Is there a concrete example of this working from the sources?
Yes, a really prominent example mentioned is the work by Stokes and colleagues. They used a type of ML called deep learning, which is particularly good at finding subtle patterns in very large data sets. They used their deep learning model to screen a massive chemical database called ZINC15,
Okay.
and their model identified several potential antibacterial compounds. One notable discovery was a molecule they named halicin. What was exciting about halicin was that its chemical structure was completely different from any known classes of antibiotics.
Wow, so the AI found something structurally novel, not just a variation on an existing theme.
Exactly. It demonstrated the power of these unbiased computational screening approaches to discover genuinely new chemical scaffolds that could potentially be developed into new drugs, helping overcome resistance to existing drug classes.
That's incredible potential. What about designing new molecules? The sources mentioned antimicrobial peptides, or AMPs.
Right. That's another exciting area where ML is making contributions, especially in designing novel therapeutic molecules. First, what are AMPs?
Antimicrobial peptides, AMPs, are short chains of amino acids, essentially small proteins. They're part of the natural innate immune system in many organisms, including us. They often have broad activity against bacteria, fungi, and sometimes viruses, often by disrupting their cell membranes.
Okay, so natural defenders. Why are they particularly suitable for ML approaches?
There are a few reasons. First, their activity is often strongly related to their amino acid sequence and the resulting physical properties, like overall charge, structure, and how much they like or dislike water. This sequence property relationship is something algorithms can learn relatively easily. Second, they're typically quite short, maybe less than 30 or 50 amino acids long. This makes them computationally more manageable to model and, importantly, easier and cheaper to actually synthesize in the lab to test the predictions compared to large, complex proteins or intricate small molecules.
So ML can learn the design rules connecting the amino acid sequence to antimicrobial activity and then potentially generate new sequences predicted to be active.
Exactly. It can be used for de novo design. For instance, one study mentioned by Wu et al. Used ML to design a completely new 12 amino acid AMP, which they call DP7. They trained their model on known AMP sequences, synthesized the designed peptide, and found it had potent activity against nasty bacteria like Staphylococcus aureus, including antibiotic-resistant strains, MRSA, and it even worked in animal infection models.
So they computationally designed it, then proved it worked.
Yes. Another example involved Porto et al., who used a technique inspired by evolution called a genetic algorithm combined with ML. They used it to design optimized AMPs based on templates found in plants, leading to the discovery of a novel peptide called guavanin-2 with broad activity against various pathogens.
So even though these examples are focused on bacteria, you could imagine applying similar ML design principles to create peptides specifically targeted against viral proteins or processes.
Absolutely. As our understanding of crucial viral targets and mechanisms improves, partly fueled by the computational analysis we discussed, those insights can feed into ML models to guide the design or discovery of new antiviral compounds, whether they're small molecules like halicin or designer peptides like DP7.
This provides a really compelling overview of how AI and ML are tackling resistance from multiple angles: prediction, understanding biology, finding new treatments. Now, let's zoom in on a specific recent development mentioned in our sources. There was news about scientists at IIT-BHU in India developing a new computational tool. What can you tell us about that?
Yes, that's a very interesting piece of news. Researchers at the Indian Institute of Technology, BHU, in Varanasi, have developed what they describe as an integrated, multi-scale computational framework. What does a phrase like computational framework suggest to you?
Framework usually implies something more than just a single piece of software. It sounds like a structured collection of different computational tools, maybe models, methods, data processing steps, all designed to work together for a specific purpose. And multi-scale suggests it operates at different levels of detail or complexity.
That's exactly right. It's an integrated system that brings together several sophisticated computer-based techniques. The sources mention it combines things like advanced methods for protein design, machine learning algorithms, and also very computationally intensive simulation techniques like hybrid quantum mechanics, molecular mechanics, QMMM, and other multi-scale simulations.
QMMM and multiscale simulations, those sound pretty advanced. Can you give us a flavor of what they do in this kind of biological research?
Sure. So hybrid QMMM methods are a clever way to study really large biological molecules like viral proteins. The idea is you treat the most important chemically active part, say the few amino acids right where a drug binds or where resistance mutation occurs, using highly accurate but very computationally demanding quantum mechanics calculations. Then you treat the rest of the large protein and its environment, like surrounding water molecules, using much faster, less detailed molecular mechanics or classical physics methods.
So high accuracy only where you need it most.
Exactly. It makes calculations feasible for large systems. Multi-scale simulations then try to bridge different levels. They might connect these detailed atomic interactions to larger-scale movements or conformational changes of the protein, or even link molecular events to cellular-level processes, simulating behavior across different time and length scales.
Wow, okay. That sounds like a really powerful way to get a detailed picture. What's the main goal of this specific integrated framework developed at IIT-BHU?
Its primary aim is to provide a deep, mechanistic understanding of how viruses evolve drug resistance. By combining all these powerful techniques, the framework is designed to pinpoint specific mutations in viral proteins and then simulate and analyze precisely how those mutations alter the protein structure, function, and interaction with drugs, ultimately leading to resistance.
And have they applied it to a particular virus initially?
Yes, the reports indicate that the initial focus of their work using this framework was on studying resistance mechanisms in the SARS-CoV-2 virus, the virus responsible for COVID-19.
A very timely focus. But is the framework limited only to SARS-CoV-2?
No, and that's a really key point about its potential. The researchers intentionally designed it with a modular structure. This means it's built-in components, making it flexible and adaptable. The plan is definitely to apply it to study resistance in other important pathogens and infectious diseases beyond just COVID-19. This adaptability makes it a potentially very valuable tool for broader public health research.
That's great to hear. Do we know who led this initiative at IIT-BHU?
Yes, the work was led by Dr. Aditya Kumar Pati, who heads the Laboratory for Computational Biology and Biomolecular Design, or LCBD, at IIT-BHU.
And what about access? Is this tool going to be available for other scientists to use?
That's another really positive aspect. Dr. Pati and the team have stated that they plan to make this powerful computational framework freely accessible to the global research community. This kind of open science approach is fantastic because it can really help accelerate research into drug resistance worldwide.
That's crucial for tackling a global problem like AMR.
Absolutely. And they also noted that this initiative aligns well with major international efforts to combat antimicrobial resistance and also supports key public health programs within India itself, like the Integrated Disease Surveillance Program, IDSP, and the National Program on Containment of Antimicrobial Resistance, AMR. It provides a powerful new tool for understanding the very resistance these programs are trying to track and control.
Has the work received recognition within the institution?
It has. The director of IIT-BHU, Professor Amit Patra, publicly highlighted this framework as a strong example of how India's scientific institutions are taking a leading role in applying cutting-edge research and technology to address really critical global public health threats like drug resistance.
It really does sound like we're moving into a new phase in this ongoing battle. We've gone from painstakingly identifying resistance mutations one by one in the lab to now having these incredibly powerful computational tools fueled by AI and advanced simulations that can predict resistance, uncover hidden mechanisms, design new drugs, and give us this atomic-level view of how viruses are evolving.
It certainly feels like a significant shift. The technology is providing unprecedented capabilities. Although I imagine the field, especially applying these very cutting-edge AI and simulation methods to antiviral resistance, is still relatively young.
It is, yes. Compared to decades of traditional virology and pharmacology, the application of these specific advanced computational approaches is newer, but the progress is incredibly rapid and the potential seems enormous.
So just to recap the journey we've traced, we started with understanding the basic biological tricks viruses use mutating key enzymes like TK-UL97 DNA polymerase. Then we saw how challenging that makes clinical management. Now we've explored how complex computational models, especially ML, can take raw genetic data and predict resistance, how interpretable ML can go further and find subtle biological drivers like those metabolic shifts, and how AI can even help discover or design entirely new molecules. And sophisticated frameworks like the one from IIT-BHU are pushing the simulation boundaries to get that really fine-grained mechanistic understanding.
That's an excellent summary of the progression. But as with any rapidly advancing frontier, especially one relying heavily on computation and data, there are still significant hurdles and needs to ensure we fully realize this potential.
What are some of those key challenges looking forward?
Well, echoing what we touched on earlier, a persistent need is for more data, specifically better data. We need larger, more diverse, high-quality datasets that reliably link genomic information with detailed, standardized phenotypic resistance data, covering a much wider range of viruses, strains, drugs, and even drug combinations. Data is the fuel.
Right. More high-octane fuel needed.
Exactly. And then there's the ongoing challenge of interpretability. We need to keep developing ML models that aren't just accurate black boxes, but provide genuine, verifiable biological insights. Turning them into white boxes that biologists and clinicians can understand, trust, and use to make decisions or design experiments is critical.
Making the AI explain its reasoning in a way that makes biological sense.
Precisely. And for applications like antimicrobial stewardship that rely on clinical data from sources like electronic health records or EHRs, there are specific challenges around data quality, standardization, privacy, and ensuring that models trained in one hospital system actually work well or generalize to others. And finally, a crucial consideration with any powerful new technology is ensuring equity. We need to make sure these advanced tools are developed and deployed in ways that benefit populations globally, not just in resource-rich settings.
So it's not just about the algorithms themselves, but the whole ecosystem, the data, the interpretation, the clinical integration, the equitable access.
Exactly. And tackling all of this successfully requires a truly multidisciplinary approach. It's not just the computational experts. You need the clinicians who understand the real-world problems, the microbiologists, the virologists, the IT engineers building the infrastructure, probably behavioral scientists to understand how clinicians will interact with these tools, and implementation scientists to figure out how to actually integrate them effectively into routine healthcare. It's a team effort.
It really underscores that feeling of an ongoing, accelerating arms race. The viruses evolve, humans develop new drugs, the viruses evolve resistance, and now humans are bringing in AI and massive computation to understand that evolution and hopefully get ahead. It's an incredibly dynamic and frankly fascinating field to watch.
It truly is. And, every new insight, whether it comes from a painstaking experiment at the lab bench or from a sophisticated algorithm crunching petabytes of data, it brings us just that little bit closer to potentially tipping the balance in this crucial fight.
We've taken quite the deep dive today into the intricate world of antiviral drug resistance. We've looked at the specific molecular tricks viruses use, the tough choices doctors face in managing resistant infections, and explored the really exciting ways that AI, machine learning, and powerful computational frameworks like that new one from IIT-BHU are starting to revolutionize how we predict, understand, and hopefully combat this threat.
It's a complex topic, but understanding these mechanisms and the technologies being used to fight back is key to appreciating where the cutting edge of infectious disease research and treatment is heading.
Definitely. It makes you think, doesn't it?
Yeah.
Given how quickly viruses can evolve resistance, but also how powerful these new computational tools are becoming at analyzing viral genomes and predicting outcomes, could we be heading towards a future where personalized antiviral therapy becomes the norm? Imagine a scenario where, for each patient, we sequence their specific infecting virus, feed that data into an AI, and get a precise prediction of which drugs will work best and which won't, allowing for truly tailored treatment from day one. Is that science fiction, or where this path might eventually lead? Something to ponder.